Hi, it's Jennifer Isom-Schmidtke, and today I'll be talking about stress, depression, anxiety, and post-traumatic stress disorder, otherwise known as PTSD. Walter Cannon of the Cannon-Bard theory of emotion was the one who first introduced the concept of psychological stress. He borrowed it from engineering, where it was used to describe the effects of physical forces on mechanical structures. So in the case of humans, it metaphorically describes the internal psychological and physiological effects of external forces. This thing that we call stress for humans can be thought of in two ways. As something that's external, and in that case we call it a stressor, and that includes environmental conditions that make unusual demands on an organism, a human, such as a dangerous or threatening situation, the experience of failure, or bereavement. Stress can also be thought of as something internal, and if it's internal stress, we refer to it as the stress response. And the stress response is your psychological and physiological response to stressors. The second definition of stress turns out to be the most important one because researchers are beginning to realize that the effects of stress on your body have way more to do with your perception of a stressor as being negative and harmful than the stressor itself in the long run. In fact, many researchers now think that simply the perception that stress is harmful to you in the long run can lead to death. Historically, when stressors appeared, they were infrequent and they were resolved quickly. And in that case, the stress response worked really well. For example, if you were faced with a lion on the African savanna, the stress response would kick in and it would enable you to run quickly for safety or to have enough energy to fight off that attack. And it would be over relatively quickly. And afterward, you would either not be around anymore or you would be returning to a calm and relaxed homeostatic state relatively quickly. However, our modern world presents stressors potentially multiple times a day, and these stressors can often last for days and weeks instead of minutes. So as a result, our stress response has become almost chronically activated. It really rarely has a chance to return to that calm and relaxed homeostatic state, and this can lead to serious health consequences. I'll talk about those next. So when faced with a stressor, our bodies react with a very adaptive and what's supposed to be a temporary response. And you can think of the stress response as having two complementary parts. So one has immediate effects, and the other takes a couple of minutes to kick in, but it is longer lasting. Now both parts are set in motion by the hypothalamus at the same time. So initially, the amygdala reacts to a stressor by alerting the hypothalamus. The hypothalamus gets the message and, in turn, activates the sympathetic nervous system, the mechanisms that we need for fight or flight to protect and ensure our survival. In fight or flight, the nerve endings of the sympathetic nervous system release epinephrine and norepinephrine. So epinephrine is released by nerve endings in the adrenal gland, and norepinephrine is released by sympathetic nerve endings in the rest of the body. Now, these two neurotransmitters provide immediate and quick action to meet our stressor and to engage in the fight or flight response. Here's a visual of the nerves and the two branches of the autonomic nervous system, the parasympathetic and the sympathetic branches. The sympathetic branch on the right is the one that is activated during the stress response. So when your sympathetic nervous system is activated, your heart will accelerate. The bronchi of your lungs will dilate so that you can get more oxygen into your bloodstream. Glucose is made available for energy, and epinephrine is released from the adrenal cortex, and norepinephrine is released from the other sympathetic nerve endings to speed your responses. Now, after the stressor is over, the parasympathetic nervous system, the one on the left, kicks in to calm and relax and to bring you back to homeostasis. The second part of the stress response, the part that is a bit slower to kick in but has a longer lasting role, is part of the Hypothalamic-pituitary-Adrenal axis, also called the HPA axis. This part of the stress response occurs when the hypothalamus releases corticotropin-releasing hormone, also called CRH. This gets to the pituitary gland, where it causes the pituitary gland to release adrenocorticotropin hormone, also called ACTH. Now that travels through the bloodstream to the adrenal cortex, which is also referred to as the adrenal glands, and it takes about 10 to 15 seconds. When it gets there, it causes them to start releasing glucocorticoids. The most famous of these is, of course, cortisol. So these glucocorticoids begin working more slowly. It takes minutes for them to be released, but they last much longer than epinephrine and norepinephrine. They last a couple of hours. The hypothalamus also releases at least 30 other stress hormones, including vasopressin, oxytocin, and prolactin. The adrenal cortex secretes glucocorticoids in response to a stressor as part of the HPA axis. But what are these? As I mentioned, one of them is cortisol, but there are others, and they are steroid hormones, and they act during stress in a way similar to epinephrine, but they do it more slowly, as I mentioned before, than epinephrine, and they last longer. One of the important things glucocorticoids do is increase the amount of calcium that enters neurons. Now recall that neurons communicate by releasing neurotransmitters into a synapse where they cross over to the postsynaptic neurons' receptors to bind with those receptors and communicate that signal. But the amount of neurotransmitter released by the presynaptic neuron is dependent on the amount of calcium that is able to enter that presynaptic neuron to free up those neurotransmitters. Now, glucocorticoids increase that amount of calcium that's available. So they increase the amount of neurotransmitter that's released, and this helps to produce extra vigilance and arousal during stress. The epinephrine and norepinephrine work more effectively because there's more of it. The last thing that glucocorticoids do in the stress response is they help to increase stored energy to mobilize it for use during that stressful situation. In the beginning of the stress response, when you first perceive that threat, whatever it is, it's the amygdala that's signaling that starts the stress response in motion. And it does this by signaling to the hypothalamus. It signals to the hypothalamus to start the stress response by releasing CRH. But the amygdala is not the only part of the brain that can exercise some power over this stress response in the HPA axis. Remember, the brain, especially the limbic system and its emotional parts, is composed of many, many feedback loops, and those loops help to regulate emotional responses and emotional memories. The hippocampus can also regulate the HPA axis. It has receptors for glucocorticoids, so it's part of that feedback loop. It can signal back to the hypothalamus to slow down or even stop the release of CRH if there's too many glucocorticoids. The autonomic nervous system can also influence the functioning of the HPA axis and the stress response. There is sort of an anxiety feedback loop, such that the amygdala gets the sympathetic nervous system going, and then the aroused and activated sympathetic nervous system can feed back to the amygdala. So the anxiety, in a way, feeds on itself. On the other hand, the parasympathetic nervous system, the relax and restore part of the autonomic nervous system, can also exercise a little power and control as well. It can work to deactivate the amygdala and reduce the anxiety. So these are just a few of the circuits that can exercise control over that HPA axis. As I mentioned before, the stress response worked great for humans long ago because when the threat was a temporary one, like running from a stampeding elephant, as soon as the threat was over, assuming you escaped, of course, the parasympathetic nervous system would come online, calming you down and returning you to your normal homeostatic state. So the stress response was positive and adaptive. That increased adrenaline made you run faster or scream louder or fight harder, and this made it easier for you to survive. The glucocorticoids worked to help conserve and create energy for the important task of eliminating that stressor. But the stressors of our age are not the same as they experienced in the African savanna. They tend to be much more frequent and last much longer, and as a result, our stress response has become chronically activated. We are really not well equipped for modern stressors. And as a result, chronic stress activation has had some really serious effects on health. For example, increased blood pressure as a result of sympathetic nervous system activation has led to higher risk of heart disease and stroke. The walls of your blood vessels are continuously stressed and stretched from higher pressure of blood. When your blood pressure goes up, it creates a higher pressure and it stretches those walls. And they begin to develop microscopic tears. So immune cells begin to fill those tears. And higher levels of epinephrine that are released during the stress response tend to increase the clotting of blood cells. That, combined with cholesterol circulating in your blood, begins to plug up those tears with gunky plaque, making it even worse. Eventually, you have clogged arteries. Chronic stress also leads to exhaustion, mood disruptions, and potentially depression, not to mention anxiety, and it also compromises our immune systems. We know the hippocampus is important in the formation of new memories, and so as a result, chronic stress can lead to poorer learning and memory ability. So long-term stress makes you stupid. Not only that, long-term chronic stress also appears to be related to unhealthy brain aging that sets a person up for a higher risk of developing Alzheimer's disease. Some of the research findings for brain damage related to stress include the volume or size of the hippocampus has been shown to be smaller for chronically stressed Vietnam veterans. And in 9/11, people who were near the Twin Towers when the disaster struck had fewer neurons in their cerebral cortex overall. Here's an example of the effects of stress on the hippocampus in monkeys. On the left is the hippocampus of the control monkey. The blue arrow is pointing to a row of healthy neurons in the hippocampus. On the right, you can clearly see that the neurons in that same area are greatly reduced in the hippocampus of the monkey who was chronically stressed. So here you can clearly see the effect of chronic stress on neurons in the hippocampus. But not everyone responds the same way to stress. It appears that early developmental experiences have an effect on the stress responses of individuals when they become adults. For example, an early study found that rat pups who were held as pups were less susceptible to stress as adults. But later research has suggested that it wasn't that the rats were somehow protected from adult stress by being handled early in life. It may actually have been the response of the mother rat when they were returned to the nest that may have made a difference. The mother rats comforted their pups with lots of licking and grooming. It's thought that that extra attention may have caused an epigenetic effect, in that it may have caused a long-term effect on the expression of the genes related to the stress response in those rats, making them less sensitive to stress as adults. A similar phenomenon has been observed for humans. Examination of suicide victims has shown that the brains of those who were abused as children show differences in stress hormone receptors from those of non-abused children, suggesting that early abuse created an epigenetic change in areas particularly responsive to stress hormones. Also, as I mentioned before, recent studies have indicated that a person's beliefs about the effects of stress can also determine whether stress will have a harmful effect. Those who did not believe that stress was harmful to one's health did not show detrimental effects. Another thing that can buffer a person from the harmful effects of stress is oxytocin. Recently, oxytocin, which is released by touching, hugging, social interaction, sexual behavior, and even petting your cat or dog, has been shown to be protective against the harmful effects of stress, especially the cardiovascular effects. Now we'll switch to depression. Now, depression has been called the common cold of psychopathology by Martin Seligman. He's a well-known researcher in depression because it's estimated that between 5 to 20% of people will experience a major and incapacitating bout with depression during their lifespan. And rates of depression have been increasing steadily in recent years. Clinically defined depression is characterized not just by feelings of marked sadness lasting 2 weeks or more. So it's not just the blues or a temporary state of being down. It is sadness that is severe enough to have a major impact on a person's ability to engage socially and to carry out his or her regular activities, including work and play. One of the hallmark features of depression is a loss of the ability to enjoy activities that were once pleasurable for a person. Sufferers of depression often feel guilt, grief, and worthlessness, and they view the world through a cognitively biased lens of pervasive and distorted negativity. It's common for them to show psychomotor, which means movement, impairment, either retardation, so every little movement requires a great deal of effort, or agitation, which is excessive movement. It's common also to have disruptions in sleep patterns and shallow sleep, in which there's frequent early morning wakening at 3:30 a.m., 4 a.m. And people who are depressed often have elevated levels of glucocorticoids. It's been shown that more women than men suffer from depression. Now, this may reflect patterns of help seeking. Women may be more likely to seek help, and in the past, it may have seemed more socially acceptable for women to seek help than men. Or this might reflect an influence of sex hormones and the circuitry in the limbic system structures that influences rates of depression. As I mentioned previously, depression has a huge impact on sleep and the sleep stages. The depressed person falls asleep faster, spends little to no time in stage 3 sleep, and enters REM for the first time much faster than a non-depressed person. And again, they often wake frequently. Interestingly, when depressed people are awakened each time they enter REM sleep, they report a significant reduction in depression. So these abnormalities in sleep patterns are thought to be related to disturbances in circadian rhythms that are related to depression. And this is consistent with seasonal affective disorder, which is thought to occur because of seasonal variations in light that disturb circadian rhythms. So what does a depressed brain look like? Here are some of the differences that are observed between the brains of depressed and non-depressed people. Depressed people have greater blood flow to parts of the frontal cortex and the amygdala, but less blood flowing to areas in the frontal lobes involved in attention. Also, the cortex of the right hemisphere has been found to be thinner, and the hippocampus on both sides has been found to be smaller than non-depressed people. Depressed people also show abnormalities in the activity and the functioning of several limbic system structures in the brain. And Davidson and colleagues found that depressed individuals show more activity measured by EEG and functional MRI in the right frontal hemispheres compared to the left than non-depressed people. And you'll recall that this is consistent with what he found for the experience of negative affect, negative emotion. Those individuals who are experiencing negative emotion also show greater right frontal than left frontal activity. So what are the causes of depression? Well, research involving twins has shown that genes play a significant role in who develops depression. But even though it's a significant role, it's not a strong influence. And the genes that are involved have yet to be conclusively identified. Other influences, environmental factors, have been shown to be important. Higher rates of depression and mental illness for children whose mothers were pregnant during the Dutch hunger winter in World War II suggest that prenatal factors may also play a role in the development of depression. It's possible that they create a vulnerability to the disease. It is possible that some prenatal conditions create a greater vulnerability to the disease. Robert Sapolsky also points to stress as a major contributing factor. Stress has been shown to be a trigger for depressive episodes. And as I mentioned before, depressed people often have higher rates of glucocorticoids, specifically cortisol. Sapolsky argues that it's impossible to understand either the biology or the psychology of major depression without recognizing the critical role played in this disease by stress. Other theories of depression have identified potential biochemical causes. One of these is the serotonin hypothesis. The serotonin hypothesis suggests that depression is a result of not enough serotonin available in the brain. So drugs such as selective serotonin uptake inhibitors, SSRIs, which you are no doubt familiar with by now, work to slow down the reuptake of serotonin in the synapses, leaving more of it in the synapse to keep sending the message to the postsynaptic neurons. And this helps to overcome a shortage of serotonin. So an SSRI you've probably heard of is Prozac. So Prozac works by stopping the serotonin transporters on the neurons that actually grab the serotonin and suck it back into that presynaptic neuron to be packaged up into the vesicles and used again. So a lot of people who have depression have been helped by SSRIs, but it doesn't work for everybody. And the serotonin hypothesis has been tested many times, but the results are inconclusive. So many studies haven't found that depression is related to having enough serotonin. And other drugs like Wellbutrin, another antidepressant, which increases the amount of dopamine and other neurotransmitters in the synapse, such as norepinephrine, have also been shown to relieve depression symptoms. Another problem with the serotonin hypothesis is that there's often a lag of several weeks between the time the patient takes the SSRI and when the depression symptoms begin to abate, and sometimes patients even feel worse before they feel better. If the problem is simply too little serotonin, then the temporary worsening of symptoms as soon as more serotonin is available doesn't quite make sense. There's one additional, more troubling issue with SSRIs to treat depression, and that's that the use of SSRIs for children and teens for depression is related to higher rates of suicide while taking those drugs. So others have suggested that it's not just serotonin levels that lead to depression, but other monoamine neurotransmitters as well, including norepinephrine and dopamine. Using this idea, treatment is focused on reducing the amount of the enzyme that breaks down all of these transmitters in the synapse to inactivate them so they don't continue to bind with receptors and stimulate the neurons. So you'll recall that the way the communication between neurons is stopped at the synapse is either by reuptake of that neurotransmitter that's communicating across the synapse or by breaking down that neurotransmitter in the synapse by an enzyme. So the enzyme in this case would be monoamine oxidase because it would be breaking down the monoamine neurotransmitters. And in fact, drugs that were designed to stop the action of the monoamine oxidase enzyme were the first antidepressants. One really interesting fact about antidepressants in the treatment of depression is that their use has actually been shown to grow or regenerate neurons in the hippocampus of depressives, which has been shown to be atrophied. So when they take SSRIs and antidepressants, the hippocampus actually becomes larger, actually increases in volume. And if you give those individuals a drug that blocks the growth of new neurons, also called neurogenesis, from happening, the improvement in depression is also blocked. Now, other than drugs, there have been more radical treatments for depression, such as electroconvulsive shock therapy, or ECT. In ECT, the person is anesthetized and muscle relaxants are given to avoid injury from seizures that result from the electricity applied through the electrodes on the head. The exact mechanisms that make ECT an effective treatment are not known, but it's thought that it increases individuals' responsiveness to dopamine and norepinephrine. And it also appears to stimulate neurogenesis, or the growth of new neurons in the hippocampus, just like antidepressants do. So another promising treatment for depression that is less painful and less invasive comes from a relatively new technology in neuroscience called transcranial magnetic stimulation, or TMS. And this uses magnetic fields to stimulate nerve cells in the brain. So it involves placing a magnetic coil, shown in this picture that looks like a sideways number 8, near the scalp and using an electromagnet to create magnetic currents that stimulate neurons. Now, these magnetic currents can last anywhere from a few seconds to 90 minutes, and they stimulate those neurons. It's painless, and it doesn't require surgery or anesthesia, and it has been shown to be an effective relief from depression. Studies that have investigated this have applied TMS to the frontal lobes, both the left and the right frontal hemispheres, Both have been shown to relieve depression symptoms. It doesn't matter which side the magnetic stimulation is applied to. Ketamine is another very newly developed treatment for severe depression, especially depression that is drug resistant. It's a veterinary anesthetic that actually is known as Special K as a street drug. But injections of ketamine have been shown to relieve severe depression in a matter of hours. And it's thought to do that by targeting glutamate in the brain. Another interesting depression treatment is deep brain stimulation, and that's when a surgically implanted electrode, or more than one electrode, in the brain is attached to a pacemaker-like device that delivers small pulses of currents to parts of the brain important in depression. So one of the places that electrodes are being tried out is in the cingulate cortex, just above the thalamus. And this is a place that is thought to inhibit sadness. So they've put electrodes in that part of the brain and stimulate it, and they've had success in treating depression. And it's thought that the success is because stimulating that part of the brain tells the brain to stop being sad. One recent study found that deep brain stimulation in the cingulate cortex was effective for 60% of the people with drug-resistant depression. And with the other 40%, they speculate that they just may need to find another area of the brain that needs to be stimulated to have that kind of success. Cognitive behavioral therapy has also been shown to be an effective treatment for depression, and especially when it's used in conjunction with antidepressants such as SSRIs. And finally, a last treatment for depression that is simple, relatively painless, and free, and that also has been shown to be as effective as antidepressants, is exercise. Studies have found that a 30-minute brisk walk or jog 3 times a week produced relief from depression symptoms that was equivalent to taking antidepressant medication.